Hello everyone, my name is Matthew Kaldawi and this is the phototransduction in the rod cells of the retina. This right here is a rod cell, and a rod cell is two of the main cells inside the eye. Now the rod cell is turned on when light is not present, and the rod cell is turned off when light is present. Right up here in the upper left hand corner we have a photon, and a photon is a particle of light. When this photon hits this rod cell right in here, it causes the phototransduction cascade. Now let's blow up this picture up here in the discs. Now the disc, we have a blown up version over here, and we want you to pay attention to this negative 40 millivolts right over here, because that plays an important key role later in the next slides to come. But we're going to focus right here on these tiny little purple pigments, which again we blow up over here, and this purple pigment itself is called rhodopsin. Now rhodopsin is made up of two proteins, one opsin and one retinal, and this rhodopsin molecule is an intermembrane protein that stretches through the phospholipid bilayer. Right, now moving on, the 11 cis retinal, as you can see, is the squiggly portion when it is in the dark. But when this photon right here hits it, it causes it to squiggle out into a straight line, and now it is called 11 trans retinal. With this happening, this rhodopsin molecule is now attracted to this molecule called transducin. Now transducin, it carries two portions. There's an alpha subunit and then there's a phosphodiesterase. When transducin binds with the opsin, this causes the transducin, or the GDP portion of transducin molecule, to catalyze into GTP, and GTP breaks off the transducin molecule and binds with the alpha subunit. This causes the alpha subunit to bind, causing the phosphodiesterase to activate. As we move on, the PDE converts. The important role with PDE is because it converts cGMP into GMP. Cyclic GMP is as you can see, it's this green little molecule right here, and what it is used to do is to keep these sodium gated channels open, but without it, and this GMP raises inside the cell, it closes these sodium gated channels. And how it does this is it synthesizes guanylyl cyclase, which is called protein kinase G, which is a lysis enzyme that breaks down cGMP into GMP. When there is no light, cGMP dominates the rod cell, keeping these open, and when there isn't light, when there is light, the channels are closed. Okay, so without this cGMP inside the rod, the sodium channels are closed, and the membrane starts to hyperpolarize toward a negative 70 millivolts. Now that's important because it causes the rod cell to turn off, and as you can see, there it is, the negative 70 millivolts right up there in the upper left hand corner. And the reason why it goes to this is because there's a drastic change due to the outward potassium current, and these are non-gated, so they're always flowing in and out, and potassium naturally likes to go towards a negative 70 millivolts when inside the membrane. And when this rod cell is turned off, it stops signaling neurotransmitters towards this bipolar cell. And as you can see, there's no neurotransmitters because the sodium is not going in. It's hyperpolarized, so this rod cell is completely turned off. But what happens with a bipolar cell is it turns on. This sends a signal to the brain telling the brain that there is light in the eye. So now we're going to go to when the rod is in a resting state. The resting state is it's starting to get back to this negative 40 millivolts. It's dark current, so there's no light in the eye whatsoever, and there's neurotransmitters being released down here. So as you can see, the sodium channels are open, causing this to depolarize back to that negative 40, and there's an increase in calcium ions inside the cell, and this signals the release of glutamate neurotransmitters right down here. And this turns off the bipolar cell. So this bipolar cell is turned off, which is really interesting because usually when neurotransmitters hit something, it turns something on. But in this case, it's turning it off, telling the brain that there is no longer light, or there's no longer light in the room. So the rod cells are needed at this point. And how it gets down to that negative 40 millivolts is it has this sodium, potassium, and calcium pump. And it's basically used to rid the system of this calcium, because there's a rapid intake of calcium. It's going to cause the the membrane to rise above the negative 40 millivolts, and you don't want that because you want it as resting state. And as we move on, here's an overall synopsis of the process. We have a light here, and it hits this rhodopsin molecule, and this rhodopsin attracts this transducin. This transducin causes the PDE to activate, switching cGMP in the cell to GMP. And this cGMP, which is keeping open these sodium channels open, is going to start to fade away, and we're no longer going to see these sodium channels open. They're going to start to close, and as you can see, this is in the dark, and this is in the light. And this is how the phototransduction cascade works.